this PowerPoint, we're going to talk about how organisms regulate their internal environment. Specifically, we're going to focus on one environmental condition, osmolarity, meaning how concentrated is the blood, or what is the water concentration of the blood or of the organism. So there are really two schools of how internal environment is regulated. We have those that do regulate their internal environment and maintain constant internal conditions called homeostasis. And then we have organisms that are conformers. They conform to the external environment. So they allow their internal conditions to fluctuate along with the external changes. So an example of this, thermoregulation. We have organisms such as mammals, in this case a polar bear, that are regulators. They're warm-blooded. They maintain a stable internal temperature. But then we also have organisms that are cold-blooded, and these are known as conformers. So things like reptiles and insects, where they take on the same temperature or similar temperature to their environment around them. With osmoregulation, which is going to be the main focus of our PowerPoint today, we do, again, have organisms that are regulators, so things like fish that actually regulate their osmolarity, meaning how much water is in their body. And we also have conformers that take on the same concentration of water and salt around them. So homeostasis is keeping a balance. We've talked about this before. Way to think about it is home status. Keeping balance or keeping a set of specific conditions within the internal environment of the organism. And some of these things that we have to maintain at homeostasis are things like temperature, blood sugar levels, energy production, water balance, which is going to be the focus of this particular PowerPoint, nutrients, ion balance, and cell growth. Maintaining a steady state is what is known as homeostasis. So regulating the internal environment with regard to osmoregulation, meaning maintaining water balance and the removal of nitrogenous waste. So in the beginning, all living organisms were single-celled organisms. And as a single-cell organism, diffusion was more than adequate for bringing materials into the cell and removing materials out of the cell. But as organisms became more complex and stopped being single-celled organisms and grew to be multicellular, diffusion stopped being adequate enough. And so therefore, organisms evolved an internal system to move materials throughout the organism as well as remove waste. Diffusion was just simply too slow. So we created, through evolution, a system of exchange. So we have a way to distribute nutrients, so our circulatory system will distribute nutrients throughout our body. And we also created a system for removing waste, and this is known as your excretory system. So the system's to support multicellular organisms. Osmoregulation is referring to water balance. So we have organisms that live in fresh water, meaning that their environment is hypotonic to their cells. And with these organisms, they have to control and prevent water from flowing into their cell and from losing salt. Saltwater organisms, their environment is hypertonic to them, and so they're always in danger of losing water, so they have evolved systems to help them maintain water. And then on land, it's a very dry environment, which means the organism needs to conserve water, meaning prevent water loss to the air around them. So why do all land animals have to conserve water? Well, for one, in living in land environments, we're always losing water. Every time you take a breath, you're losing water. Every time you remove waste from your body, you're losing water. And we may lose life while searching for water. Back in the days when we were all cave dwellers, searching for water was a dangerous pastime. So intracellular waste, what is it that we produce as waste products? So when you eat carbohydrates and lipids, you produce, or you break down your carbohydrates, you break down your lipids, and you produce carbon dioxide and water. Both of these things are easily exhaled from the body. Water is reabsorbed into the body and really aren't a big deal. But when you digest proteins and nucleic acids, in addition to getting the carbon dioxide and water, you produce nitrogenous waste, which unfortunately is pretty toxic to the body. This is why you produce nitrogenous waste. Most of the nitrogenous waste comes from the breakdown of proteins, specifically the amino acids. And you can see here's our amino acid. So the central carbon group and our carboxyl group are what's going to form our carbon dioxide and water. But the amine group is what becomes our nitrogenous waste and forms ammonia, which is extremely toxic to living organisms. So we've got to get rid of this ammonia. So ammonia, very toxic. Here's our spinning ammonia molecule there. And carcinogenic, meaning cancer-causing. Ammonia is also very soluble, so it easily crosses membranes and gets into cells it's not supposed to. So we need to get rid of this ammonia as quickly as possible. Dilute it with water and get rid of it fast. So how do you get rid of nitrogenous waste? Well, that depends on what type of organism you are and where you live. So whether you're aquatic or terrestrial or an egg layer. So nitrogenous waste for aquatic organisms, because they're surrounded by water, they can afford to lose lots of water. So simply we take the ammonia, dilute it with lots of water, and excrete it through the skin of the fish. Terrestrial organisms, we need to conserve water. So what we do is we convert that ammonia to a substance called urea, which is far less toxic than ammonia, and we still dilute it with a little bit of water. Terrestrial egg layers need to conserve water, but they also need to protect their embryo inside of the egg. So they take the ammonia and they produce a really large structure called uric acid, which is least toxic of all and requires as little water. So again, freshwater animals, they remove the surplus of water that would be entering the cells, mostly through the gills or moist membranes, and also overcome the loss of salts. Land animals, nitrogen waste disposal, we need to conserve water because water is not all around us. Must process the ammonia so it's less toxic. So we take the ammonia and convert it to a slightly larger molecule called urea, which is less soluble, so less likely to get into the cells we don't want it to be, and less toxic. And this substance is produced in the liver. So here is urea, and you can see that urea is really just two ammonia groups connected to a carbonyl group. So urea costs energy to synthesize because, again, this is a synthesis reaction. We're joining two ammonia molecules together to form a larger molecule. The kidney is going to then take this urea that we create and filter it out of the blood and remove it into a material that we call urine. So the yellow part of the urine, the part that gives that yellow color to your urine, is the urea that we produce, those nitrogenous waste in the form of urea. Now, if you're an egg layer, such as a reptile or a bird, you have to have nitrogen waste disposal while you're still inside of the egg. And there's no place to get rid of it because you have this itty-bitty space with a shell around it. So you need an even less soluble molecule. And what reduces solubility is the size of the molecule. So reptiles and birds convert their nitrogenous waste to a much larger molecule called uric acid. And so you've probably all seen uric acid before. Uric acid is polymerized urea, and you can see multiple urea groups there, and there on our uric acid. doesn't precipitate out of the solution, so it doesn't harm the embryo inside of the egg, forms a white dust, and adult birds and reptiles still produce this white paste with no liquids, and this is what you know as the lovely white component of bird poop. So birds don't actually pee. They only produce waste in solid form. The white part of bird poop is actually their nitrogenous waste, or what would be our equivalent of urine. So mammals, we're going to focus on mammals because that's what we are. That's what humans are. We're mammals. Mammals meaning that we're warm-blooded, we have hair, we produce our children live and whole, and really the mammal part comes from the fact that we produce milk with something called a mammary gland. So mammals, our filtration system are our kidneys. And so in humans, we have two kidneys. These guys right here are the kidneys. And those kidneys are connected via tubes to our bladder, and then the bladder collects our liquid nitrogen waste and removes it from our body. Within the kidney, there are these smaller units called nephrons, and there are millions of nephrons per kidney. And it's the nephron that is the actual filtration unit that removes the waste from our blood and out of our body. So in the mammalian system, the kidneys clean the blood. What they're going to do is they're going to remove waste and excess solutes, and they form a material called filtrate. Filtrate is all the material that was removed from the blood by the kidney, and then this filtrate collects and forms urine. What returns back to the blood is any water or needed solutes. So really what happens is the kidneys remove everything from the blood first, and then they selectively put things back as needed. So if you're feeling dehydrated, the kidneys will actually add more water back to your blood. If you're low on salt, same thing, it'll return salt back to your blood. And this all functions because we have something called counter-current exchange between the circulatory system, which is indicated here in red, circulatory system meaning moving our blood, and the excretory system. So here in yellow, the filtrate is the material that was removed initially from the blood that's going to be flowing through our excretory system. And we call this counter-current exchange because the blood and the filtrate actually travel in opposite directions of each other. And the result here is that we end up with blood with just the right amount of solutes and water and a filtrate that becomes the urine. The main functions of the kidneys and the mammalian excretory system is one, filtration, meaning remove water and solutes out of the blood and waste. And then reabsorption. As the filtrate moves through the excretory system, materials are going to be selectively reabsorbed as needed. So, for example, again, if you are dehydrated, we will reabsorb more water. If you're low on salt, we'll reabsorb more salt. That would be the solutes. Secretion, pump out any other unwanted solutes to the urine. So anything that remained in the blood that we want to get rid of, we pump out. And then excretion, we actually expel the concentrated urine, so the nitrogenous waste, excess solutes, excess water and toxins from our body in the urine. So mammalian kidney actually is the point of interaction between our circulatory system and our excretory system. So the circulatory system is made up of something called a glomerulus. So this little tangled ball of little red tubes is called the glomerulus. And the glomerulus is made up of really tiny vascular tissue called capillaries. And here what's going to happen is the blood from the body is going to enter into the glomerulus, and all the materials are going to be filtered out of the blood and into what's called the Bowman's capsule, which is the start of the excretory system. And then whatever was left in the blood is going to travel this way down through and around the nephron. The excretory system consists of the nephron. Um, the nephron starts with the Bowman's capsule, which is right here, and is followed by the loop of Henle, which starts with proximal tubule, descending limb, ascending limb, and distal tubule, and then finishes in an area called the collecting duct. So how can different sections allow the difference of different molecules to pass through? That actually has to do with permeability of this tube. In some places in the tube there are transporters for water, in other places of the tube there are not transporters for water. In some places along the tube there are sodium pumps that are going to pump sodium, but in other parts of this tube those pumps are missing. So looking at kidney structure, again, what you need to take a look at, here's our full kidney, and then all these little lines here represent nephrons. There are millions of nephrons that make up a single kidney. So here's a nephron, the functional unit of the kidney. This is actually where filtration takes place, and so the nephron is all this part here in tan. The part here in red and blue represents actually the circulatory system in your blood. So there's about 1 million nephrons per kidney, and the function of these is to filter out urea, so the nitrogen waste, and other solutes, so excess salt, sugar, etc. Blood plasma is filtered into the nephron, so blood plasma is the liquid component of your blood, and this happens because there's actually a pressure gradient. So the materials are actually being pushed out of the blood via the fact that there's this high pressure of the blood and lower pressure of the filtrate. And then what happens in the nephron is we get selective reabsorption of valuable solutes, so things like sugar and salts, and water back into the bloodstream as needed. And this occurs because we have countercurrent flow, so the filtrate, the materials in the nephron, flow in one direction and the blood actually flows in the opposite direction. And this forms something called a countercurrent exchange. So taking a look at the nephron structure. Here's the kidney. Zooming into the kidney, the middle part of the kidney is called the medulla, and the external part or the outer layer of the kidney is called the cortex. The medulla is very salty. This is where we're going to have a high sodium concentration, and the cortex is much lower in salt concentration. Here we have the Bowman's capsule. The Bowman's capsule is the start of the nephron. Within the Bowman's capsule we have the glomerulus, which is a cluster of blood capillaries. After the Bowman's capsule, we have the proximal convoluted tubule. Proximal meaning before, convoluted meaning twisted, and it's before the loop of Henle. So proximal meaning the twisty tube before the loop of Henle. Here's the loop of Henle, and the loop of Henle consists of the descending limb, meaning the down limb, and the ascending limb, meaning the up limb, and then the distal convoluted tubule, distal meaning after, or the portion that comes after the loop of Henle. And then finally, the last part here is called the collecting duct. The collecting duct is actually separate from the rest of the nephron. So what happens at each part? So the glomerulus, we filter out water, glucose, salt ions, and urea from the blood, and this is done due to a pressure gradient. So pressure is actually pushing these materials out of the blood, and it forms a material called filtrate. Filtrate meaning the materials filtered out of the blood. And it's the filtrate that's actually going to move its way through the nephron. What's not filtered out are going to be cells and proteins because they're far too large to make it across the capillary membrane. Okay, so again, if we take a look here, this is the blood. This is the membrane between the blood vessel and the Bowman's capsule, and you can see things like water, salt, um, glucose, and urea are going to be moved from the blood and then into this part here called the filtrate. So the proximal tubule, um, again is this twisty part before the loop of Henle. We're going to reabsorb materials back into the blood. So we're going to reabsorb some salt, we're going to reabsorb some water, some glucose, and then also bicarbonate. And bicarbonate is used to buffer the blood or keep the blood pH constant. So within the proximal tubule, there's actually a bunch of different protein channels that transport the materials from the filtrate back into the blood. We will have some facilitated diffusion, so things like water will be transported by facilitated diffusion. Glucose will be transported by facilitated diffusion, and then salt will actually be actively transported by a sodium pump. So the loop of Henle has two parts. The first part is called the descending limb, and it's highly permeable to water. And this is because there's lots of aquaporons, little protein channels that transport water via facilitated diffusion out of the filtrate back into the blood. Additionally, the descending limb has low permeability to salt, meaning that salt is not going to be able to cross this membrane. And so at this point, water is going to be reabsorbed back into the blood. The other side of the loop of Henle, known as the ascending limb, meaning the up limb, has low permeability to water, meaning water cannot transport through the membrane of the ascending limb. This membrane also contains chlorine pumps, which cause sodium to follow by diffusion. And so in this part of the limb, salts are reabsorbed back into the blood. And it's this structure that actually helps maintain osmotic gradients. So as salt is being pushed out into the blood and the blood is becoming saltier, as the blood travels the other way around to this side of the loop of Henle, water is then going to diffuse back into the blood because of the concentration gradient between the solutes that are going to be in the blood and the solutes that are in this descending limb. The distal tubule, distal meaning after the loop of Henle, we've reabsorbed salt, some more water, and then pretty much all the bicarbonate ions back into the blood. And again, bicarbonate is used to buffer the blood and maintain pH. The collecting duct, the final part of the nephron, we reabsorb water, and whatever we don't reabsorb back into the blood, so anytime I say reabsorb, meaning the material is going to travel back into the blood. Whatever isn't reabsorbed is going to be excreted from the body and pass out of the body as urine. So how is this reabsorption achieved? So what happens is there's a tight osmotic control to reduce the cost of energy. Anywhere we can, we're going to move water by diffusion as opposed to using active transport. And this happens because we use a counter current exchange, meaning that the blood flows in the opposite direction of our filtrate. So if we take a look here, here's our blood. So the blood entered in the glomerulus, and here's filtered blood. So this is blood with all the solutes removed. And so what we're going to do is we're going to put solutes back by actively pumping sodium back into the blood. So as the blood travels past the ascending limb, the blood going down is getting saltier or increasing in solute concentration. So when the blood comes around to this side and is running parallel to the descending limb, the blood is salty enough to cause water to diffuse back into the blood and be reabsorbed. So the summary, what's not filtered out of the blood are the cells and the proteins. They remain in the blood because they're far too big. What is reabsorbed by active transport are going to be things like sodium and chloride, amino acids and glucose. And what's reabsorbed into the blood by diffusion, again, there are going to be some sodium and chlorine, but mostly water. And then what's excreted, what's going to stay in the filtrate and leave the body, are going to be the nitrogenous waste, urea, excess water, excess solutes, so extra glucose, extra salts, and toxins. So why selective reabsorption and not selective filtration? It's actually easier to take everything out first and then selectively reabsorb things as needed by concentration gradients. If you have any questions, we'll reserve those for class.